Well, thank you so much for having Searchlight be part of your conference today. As mentioned, my name is Astrid Rasmussen. I'm the project director for Simon Searchlight. And I want to share with you what Simon Searchlight is, what we do and how we do it. So we're in an international research program, which is fully based online. And our goal is to generate resources to accelerate the science and improve the lives of people with rare genetic neurodevelopmental conditions. We do this by taking a genes first approach, by which we mean that to be eligible to participate in the study, participants and their families must have a variant in one of a curated list of genes that we study. And we verify such inclusion criteria by reviewing clinical testing results from around the world, currently representing 108 countries so far. And we generate high-quality resources such as data and biospecimens. And then after cleaning and collating the data, we share our findings back with research participants and their families, and also with approved and vetted scientists. And we try to establish a bridge between the two main communities, be it the researchers and the families. It is a multidisciplinary team across three main institutions: Boston Children's Hospital, where our leader, Dr. Wendy Chung, is based. Then the Simons Foundation, which is our funding agency, and the Geisinger Health System. And we receive feedback and guidance from an external community advisory committee. These are either people affected or their family and caregivers, and they keep us on track to keep in mind their needs, center of all our goals. As mentioned earlier, we are funded by the Simons Foundation. The Simons Foundation has provided 100% of Searchlight's funding for the past 16 years now. They do it through what is called SAFARI, or their Autism Research Initiative. And besides being funders, they also guide the scientific direction in conjunction with Dr. Chung. We are part of their cohorts subset of research, and we're two main cohorts: Simon Searchlight, which is the genes first cohort, and we have a companion study called Spark. And Spark is centered around diagnosis. So this is a very, very large cohort, more than 100,000 affected individuals with a diagnosis of autism, whether it's genetic or not, in basis. And combined, we generate the following resources. It is Safari Gene, which is a curated list of genes and functions that intervene in these conditions. Safari Base, which is where the behavioral, clinical, and genetic data are housed, a DNA and PBMC repository, a repository of induced pluripotent stem cells, animal models, post-mortem brain tissue, and there's also funding for research. So it's a very encompassing program. And as mentioned before, we try to work in conjunction with the other pieces of the Safari program. So, for example, subjects that enter the autism cohort Spark and receive a genetic diagnosis that is within the list of genes in Simon Searchlight can then be transferred over to our study, so we collect all the data and additional specimens from them. Which are these genes? Currently, there are 184 conditions covered in Simon Searchlight. As you can see, multiple of the genes that you are interested in the GRIN and GRIA genes are included. And we accept participation in many languages because part of our goal is to have as representative a sample of data and people as possible. So the study is conducted in English, German, Spanish, French, Italian, Dutch, and Portuguese. And the steps to be a part of this are the following. It starts up with registering online and providing informed consent, and then submitting a genetic lab report to see if you're eligible. If so, then there's sharing of medical history, completion of surveys, optional blood samples if that is something you're interested in, and then longitudinal participation with annual updates. This is how those steps look. This is our dashboard online where you start your process by registering and consenting, after which you start getting assigned different tasks. Each task is assigned with a certain cadence, and it is based both on status, so if it is an affected participant or an unaffected family member, the age, gender, and several other conditions. And once a task is completed, there are online incentives in this form of gift cards. These are also international, so anyone can receive those incentives. And you are kept abreast of what your progress is, and you can always consult your dashboard. What types of questionnaires do we administer? These are the ones that are administered for people who have genetic variants in those genes. And by that, we mean pathogenic, likely pathogenic, and variants of unknown significance. And we collect demographic and genetic data, history of all medical diagnoses and conditions, and detailed seizure history, also psychiatric diagnosis, sleep questionnaire, and then a battery of both in-house developed and publisher created measures on development and behavior. All are assigned at baseline, and then many of them are also assigned annually so we can have a natural history of the condition. Some of the most relevant surveys are our annual medical history. This is a very long survey, so the first time you complete it, it does take probably an hour or more to complete. But after that, every year, you just receive the same pre-filled questionnaire and are asked to update any changes or new things that have happened. So it's much easier to do that. And another recently launched survey is the ORCA, or Observer Reported Communication Abilities, which is a description of the language skills of the child as described and observed by a primary caregiver. So not a clinician, but what happens in real life at home. And this is specifically to gauge minimally verbal and nonverbal communication styles. The next step I mentioned was the option of donating a blood sample. You don't need to agree to a blood sample to be part of the study, and it's noteworthy that currently we can only offer this to participants in the United States due to difficulties with some regulations, customs, and the time in transit to secure the quality of the samples. But for those in the US, we, if you are interested in doing this, we send you a blood kit in the mail, help you schedule an appointment with your local Quest diagnostics lab. If Quest is not available, then we can schedule in-home blood draws through TravelLab. And once the blood is drawn, the lab takes care of the packaging, shipping, sending it back to our repository. All this is at no cost to you. And we accept from the participant and the parents and up to 2 unaffected siblings as match controls. What do we do with the blood? The blood is processed at the lab to generate whole blood DNA, sometimes lymphoblastoid cell lines, not always, but we also separate what are called the peripheral mononuclear blood cells. So those are cells, white blood cells that have a nucleus and therefore contain DNA, and those are banked so they can later be reprogrammed to create iPSCs or induced pluripotent stem cells. The current process is that there are two ways of getting to stem cells. One is we have a small program in which we sponsor the creation and the cost of creating those cell lines. There are currently a little over 200 cell lines right now available that were paid for the Simons Foundation and then Nancy Lurie Marks Family Foundation and are available for scientists to request. And going forward, there is an application process in which you can submit an application to get stem cells created. Not all will be approved because it's expensive, but if we can't approve it, and you have an interested scientist who's able to create stem cells themselves, then they can request the PBMCs and create the stem cells. And it is a model in which the stem cells that are created are some portion of them are returned to our biobank so they can be further expanded and distributed to future scientists. In terms of returning information for families, we do things as this one, which is present at conferences and meetings so you can hear what we're doing. But we also send you some information in the dashboard where you complete tasks. You can see some of the data that was collected on you and how it compares to other communities. And every quarter, we share quarterly reports with updates on the condition and either from the annual medical history survey or the progress of the Vineland questionnaire. Another way of engaging with you, which is what I mentioned about connecting researchers with patients, is we have a program called Research Match, in which we like to think of as a matchmaking service or online dating almost between scientists interested in a condition and participants and families that have such conditions. So if a scientist approaches us saying, I want to study X clinical feature or, for example, do MRIs or EEGs in participants with this specific green condition, we can say, okay, we can send out an email to all the Searchlight participants with that condition and invite them to participate in your study. In addition to doing that, we can also offer funding for the scientists, and if they don't have the resources themselves, we help them create their surveys and use our online platform to do so. We also, as mentioned, distribute data and biospecimens, and this is an updated list of materials that the overall Simon Searchlight cohort, which we're hoping this month will reach 10,000 subjects total. We're at 9,990 as of last evening. And this is what we have distributed. There have been more than 300 data requests from multiple scientists and multiple institutions. This is clinical data, genotype data, MRI and EEG from earlier stages of the study. We don't collect that currently. More than close to 1,500 requests for DNA and blood samples, 335 requests for stem cells, and 1,200 vials of stem cells distributed, DNA and lymphoblastoid cell lines, and this include green materials. And then we also distribute these materials as literature for scientists. We return findings to ClinVar, so there's updated genetic information. We allow students to use materials for their dissertations, and there have been more than 120 scientific publications derived from our data. And how do scientists get to use that data? They do it through what we call Safari Base, which is the online platform housed out of the Simons Foundation, where the scientists can request data. It is vetted and approved by a scientific committee, and then they can get access to all of this. And then I want to show you just briefly, because as mentioned, there's too much information on your conditions that I'm sending you a 100-slide PDF deck for you to review at your leisure, but an overview of what is there on GRI conditions in Simon Searchlight looks like this. Green to Be is by far the largest group. 211 people have registered. We have lab reports, and 134 have completed their medical history, and a few more have also completed additional surveys, and there are 52 specimens, and you can see in decreasing order how the other groups are doing. It is important to remember that we're longitudinal. Oh, sorry, this is going a little too fast for me. That we're a longitudinal study, so we want to continue collecting. It's not important to only register. We need you to stay with us over time so we can answer a question we hear a lot, which is, what can I expect down the road when there are teens, when there are adults, middle age, and so on? How varied are these people in the study? Extremely varied. This is how the distribution of variants looks in the Green 2B gene. As said, we have 158 people that we have lab reports for. This increased by 19 since last meeting. There are in blue loss of function, gain of function variants, in red loss of function, in black those that do not yet have been characterized from a function point of view. You can see they're all over the gene and all types of mutations. As we go down the list, there are fewer people, less variety, but still good representation. This is Green 2A, Green 1, Green A3, GRIA 3, GRIA 2, and Green 2D. All these slides are in the slide deck, so you can take your time and look at them. And then these are the derivatives of the blood samples that have been created. We have PBMCs for some people. We have induced pluripotent stem cells only for Green 2B so far, lymphoblastoid cell lines, DNA from different sources, and saliva for a few. And this is a list of variants included in the induced pluripotent stem cells. There is one line that was attempted several times, and we have run out of material, so this line will not be created. It's row 11, and there's one still in process. For those scientists that want them, they are distributed just for the cost of the shipping, which is $124, as opposed to the thousands of dollars that it would cost to make them. And they are by request to the Simons Foundation. What the clinical data look like, again, it's just a super bird's eye view of how it looks like, but we have a more or less broad distribution of ages in childhood and adolescence for the main three groups, which are Green 2B, Green 2A, and Green 1. But as you can see, very little representation of adults. So if you know of adults, encourage them to participate. Otherwise, we won't know what the natural history is. In the lower row, you can see the main developmental conditions associated with the conditions, and we can see that while all of these genes, and I'm only presenting those for whom we have at least 10 participants with full clinical data, In all of them, intellectual disability or developmental delay are the most common feature, followed by language disability. Autism is also common, and then to some other degree, ADHD, anxiety, obsessive compulsive disorders. In terms of neurological conditions beyond the developmental and intellectual disability, low muscle tone is a nearly universal aspect. And then you can see there's some coordination problems, tic disorders. Some have cortical visual impairment and coordination problems. And the gastrointestinal system is the other system that is frequently involved, be it with constipation or diarrhea, GERD, and irritable bowel syndrome.
Dr. Rasmussen, can you wrap up in about a minute?
This is my last slide.
Great, thank you.
And so why join us? Because we have 16 years now of expertise in doing this. More than 10,000 families have trusted us. It is multidimensional, international, at no cost for you. We don't charge for distributing or storing data. We don't charge for the materials. We have a community advisory committee, and we share data back to you. So, and we have high standards of security. So thank you so much for listening to me. And I hope you can review the rest of the materials so you can consider what questions you have and reach back out to us. And all our contact information is also shared in those slides.